Hey, good morning. Happy Monday. So we are continuing here with chapter 8 as a brief look forward. We are going to be finishing the material for the whole course next class, Wednesday. So today we're going to keep going with vision. We'll start what happens in our ears, hearing, and balance. On Wednesday, we'll finish the senses. We'll finish out balance, smell, and taste. Then on Friday, we will have a review session for midterm 3. And then next Monday, a week from today, on 4/27, the third midterm will open up. I am still working on the details of that endeavor. I'm hoping to have an announcement posted tomorrow about the structure of the midterm. It'll be roughly the same, it's multiple choice and short answer, but I will have that finalized by tomorrow. Okay. Then next week, so we have that next Monday, we have that midterm. The following class is a Wednesday. That's technically the last day our class meets. That will be a review session for the final. And then your final will be during finals week. Again, still have to figure out that. But stay tuned. Okay. So I'm going to switch gears and open up Canvas very briefly and to show you what you guys have coming up. Not a whole lot. We're done. Again, most of the assignments for the course are now completed. What you have is that cranial nerve worksheet, which is due tonight at midnight. And then you've got 2 last discussion boards. So in discussions, again, we've got the last week of things you found interesting or confusing. You have to post in that sometime this week, and then by Sunday. And by Friday night, you have to answer the special senses questions, the questions about this last chapter. So not a ton left to do, which is great. Okay, coming down the home stretch. Let's switch gears and talk through some more material here. As always, put a question in the chat if you have it. Okay, so last class we had talked about vision. We talked about vision, and we had spent a fair amount of time talking about what our eyes do, So this is looking at a sagittal section of a human eye, with the front being here to the left, and the brain, imagine the brain here to the right. And so if we're gonna just briefly trace that path of light from the air here coming to our eye. So how does the eye work? Well, light, again, is a wave. It's going to be coming from all directions. It's going to go through that sheer corneal layer. It's going to pass through the pupil. Remember, the pupil is an opening in your eye that allows for light to go through. And the pupil will change its shape depending on how much light needs to pass through. So if you're in a really dark room, your pupil is going to want to take in as much light as possible because it's not getting enough information. And so your pupil will be big or dilated. However, in bright light, there is way too much visual information happening. There's way too much light. So your pupil is going to constrict or get smaller in bright light. And it does this, it opens and closes, it changes its shape to maintain a constant amount of light hitting your eye, okay? That light is then going to move through the pupil and it's going to hit the lens. Now, it's helpful sometimes to think of the eye like a camera. So in a fancy, one of those big fancy person cameras, you also have the lens, and you're going to change the shape of the lens to help you focus on an object. You're doing the same thing with the lens in your eye. So your lens is going to help you with focusing. In the same way, you might change the focus on your phone for like portrait mode, something up close versus something in the distance. The lens is going to do the same thing. It's going to help focus the light to the back of your eye. So a lens in a camera focuses the light to the back of the camera where the film is, or in digital cameras where the microchip is. The lens in your eye is doing the same thing. So it's going to take the light and it's going to focus it onto the fovea centralis on your retina, in the very back of your eye. So that light wave is going to move through that vitreous humor here in the back part and hit the retina. And again, as a review from last time, in the retina, it moves in an inside-out pattern. So it's going to go all the way to the back of the retina first and hit those photoreceptors. Remember, we have two kinds of photoreceptors: rods, which are on the periphery. They see black, white shades of gray. They help you at night when everything is all dark. Cones, those are in the center for color. C for center, C for color, C for cones. You have three types of cones: blue, green, red. No yellow cones. Tell me about yellow cones, they don't exist. Okay. The photoreceptors, whether they're rods or cones, depending on what kind of visual cue you're getting, they're going to process that information, shoot an action potential, and they'll pass that on to a bipolar cell, which will pass it on to a ganglion cell, and your ganglion cell becomes your optic nerve, which leaves your eye, the optic disc. This is where we ended last time. We also talked briefly about cataracts and glaucoma. Okay. Today, let's talk about a few more facts about your eye. So eyes are made for distance vision. When your lens is fully relaxed and your eye is fully relaxed, you're able to see well into the distance, about 20 feet being the optimal distance for your eyes. When you're looking at something up close, however, you have to use those little muscles in your eyes to help you focus the lens for up close vision. And if you do this for a while, your eyes can actually become tired. This is why they recommend that every 20 minutes you take a 20-second break and look at something 20 feet in the distance. And that 20-20-20 rule will help you relax your eyes for a short period of time. And for your eyes, relaxing means staring off into space. Okay, so every 20 minutes, look up from your phone, look up from your computer, look up from your desk, stare into the horizon. This is going to relax your eyes. Your eyes, when they're looking at something up close, your lens is moving around, it's changing shape, and it's doing something called accommodation, which allows you to focus on that close object, and that can be tiring. Okay, your lens also does something interesting in that it flips and reverses the image of the world. So you're going to see the world, and your lens is going to flip it around and make it upside down and make it real small and project that image onto your retina. And so your brain has to take that information and know, okay, I've got to flip it, reverse it, and expand it. So your brain is going to take the image that it sees and it's going to process it to what it knows the world looks like. Okay. And how this information moves from your eye to your brain, we call this your visual pathway, is that the world is broken up into your visual fields. So the area in green here is showing you the area of the world that both eyes can see. And the blue area is only really seen by the left side, and the yellow area is only really seen by the right side. And so what I want you to get from this picture is that if you follow this information back from your eyes, so if you look at where the blue is, follow those blue fibers, they're going to have to cross over. So the left side of your world is actually processed by the right side of your brain. And the reverse is true. The right side of your world is processed by the left side of your brain. And that's because the information as it travels down the optic nerve is going to cross over at a spot called your optic chiasm. So the optic nerve will cross over at the optic chiasm, switching that around, right? So the left side is seen by the right side of the brain, right side of the world seen by the left side of the brain. And that path is then going to move through our thalamus, which is our relay station. Remember, it's going to say, this is visual information. Send it on back. Got to hit the occipital lobe. And it will go all the way back to the back of your brain in your occipital lobe where your visual cortex lives. And that's where the information is going to be processed. Okay. So again, visual fields are crossed over at that optic chiasm, and then they're processed by the thalamus or relayed by the thalamus and eventually processed here in our occipital lobe. Okay. Finally, let's talk about some common eye disorders that people, really cool people have. So myopia is the first one. This is more common. This is nearsightedness, where the distance is blurry. This is what I have. This is why I wear very thick glasses, because my eye has a smaller vertical axis than a horizontal axis. If you have 20/20 vision, your eye is perfectly round. The vertical axis and the horizontal axis are equal. However, if it kind of is like a grape and you smush it down a little bit and the vertical axis becomes shorter than the horizontal axis, you have myopia. Can't see at a distance. As we age, a lot of people get hyperopia, which is the opposite. It's farsightedness. This is when you can't see close up and you got to do that thing with the reading glasses or taking your glasses on and off, right? And that's because the vertical axis is now longer than the horizontal axis. Some people also have something called astigmatism, which is an unequal curvature, usually caused by a small bump in the eye, so you can't quite see. And what this means is that points will look like lines. It's a little bit of visual spreading. All of this can be fixed by glasses and contacts, and myopia and hyperopia can also now be fixed pretty easily with laser eye surgery, which is something I will never do, but people tell me works great, works great. Totally able to fix your eye. And they do this by taking a laser and slicing off bits of your eye to make it more round, change the shape. Okay, that concludes eyes. So we're now going to switch gears and we're going to talk about our ears. And your ears for this unit, we're going to identify the structure of our ears and talk about their function, detail sound localization, explain what happens in that inner ear, your organ of Corti, define hearing loss and deafness, and then we'll talk about equilibrium. And in biology, equilibrium has nothing to do with chemical reactions, and everything to do with balance. So, what is sound? What is sound? If you remember from physics, sound is a wave. It is technically a vibration propagated as a wave through a transmission medium, usually air, water. And for sound, there are two pieces of information that the wave encodes. It encodes frequency in its wavelength, or how close together those waves are. High frequency things, high pitch noises, they have wave functions that are very close together. They have a small wavelength. Low pitch noises, the waves are further apart, right? Or they have a longer wavelength. And for humans, we can hear frequencies from 20 all the way up to 20,000 hertz, hertz being the unit of frequency. The other piece of information encoded in the sound wave is the volume, and this is in the amplitude, or how tall the wave is. So things that are really loud, they have a tall wave function, whereas things that are quiet, they have a short wave function. And our ears hear both frequency and volume. So they're going to take the sound wave and they're going to turn that into action potentials that tell us about the pitch of something and the volume. So let's keep that in mind as we think about our ears. Okay, so your ear is one of the most interesting special sense organs in that it houses two senses. We'll start with hearing and then we'll talk about equilibrium. Your ear is divided into three main areas: the external ear, middle ear, which is also called our tympanic cavity, and the inner ear, which is also called our bony labyrinth. And this is what that looks like. Your external, your outer ear, is composed of the auricle. This is the thing you put earrings in, the thing that hangs off the side of your face, which leads into our external acoustic meatus, which is our auditory canal. And then you have your middle ear, which starts at your tympanic membrane. It has three small bones, three ossicles, the malleus, incus, and stapes, and that leads to your oval window, which separates out your cochlea here, and your vestibule and your semicircular canals, this bony labyrinth of your inner ear. The external ear is open to air, obviously. You got air on the outside, air in your external ear. The middle ear is an air-filled cavity as well. It's usually sealed in pretty tight there. And the inner ear is full of fluid. Okay, let's start with our external ear. So your external ear is only useful for hearing. And the structures here, we start with the auricle, otherwise known as the pinna. This is the thing you put the piercings in. And the function of our auricle is to funnel sound waves. So if sound is a wave, it's coming at you from all directions. Your pinna, your auricle, is going to funnel that wave into your external acoustic meatus. So the auricle is going to funnel the sound, and the external acoustic meatus is going to pass that sound wave and funnel it down till it hits the tympanic membrane. Your auditory canal is lined with skin and cerumeus, or wax glands, earwax. This is what happens. You are technically not supposed to put a Q-tip in there, because what you can do is you can impact the wax and build it up, and you could actually lose your hearing a little bit. Some people even have hurt their eardrum from forceful Q-tip use. So you want to keep your Q-tips to your auricles. Okay. Your external ear ends with your tympanic membrane, which is commonly referred to as your eardrum. We're going to use the term tympanic membrane. Our ears, we have two of them to help us localize where sound is coming from. So this is called binaural cues. And what your brain does is it will measure the distance in arrival time. So if a sound is coming over from your left ear is going to get that wave slightly faster than your right ear. And your brain is able to do some fun math and calculate that the sound must have been coming then from the left. And this is why if you were to snap or make a loud noise in one direction, you'll very quickly turn to that direction. Or if your eyes are closed and you hear a noise, you can usually point with some good accuracy to where that sound is coming from because you got two ears and they hear at slightly different times. Okay. Let's talk about our middle ear now, our tympanic cavity. So again, this is the air-filled cavity from our tympanic membrane to our oval and round window on the other side, only involved with hearing. And it starts with our tympanic membrane. So a sound wave is going to move down that auditory canal, and it's going to send the tympanic membrane vibrating with that same wave function. So it starts as a wave, hits the tympanic membrane starts vibrating, creating that wave function again in the air of our middle ear, our tympanic cavity. Now, usually this is a tight, sealed cavity, sealed off, only air. However, there is one kind of escape valve there, which is our eustachian tube. You'll also see it referred to as our pharyngotympanic or auditory tube. And this connects our middle ear with our throat. And normally, that tube is closed up, again, creating that tight seal. However, in times where we're experiencing a change in pressure. So for example, when you're going up in an airplane or you're going up a mountain, the air pressure is going to be lower than it was normally. And so your external ear, the outside into your auditory canal, is going to be at a lower pressure than the sealed middle inner ear. And that's going to cause your tympanic membrane to kind of bulge towards that low pressure because of that pressure difference. And that can be pretty painful. So to resolve that and to make sure you don't burst your eardrum or burst your tympanic membrane, you're going to open up your eustachian tube to help normalize the pressure and equalize the pressure in your middle ear with the pressure in your throat, which is the same pressure as the air, which would be what was in your external ear. And this is why when you're climbing a mountain or you're in a plane, you're supposed to yawn or swallow because that action will open up your pharyngotympanic tube and equalize the pressure. Little kids have much shorter tubes, which is why for them this process is much harder and their ears are much more sensitive to changes in pressure. This is part of the reason kids always cry on airplanes. So if you're on an airplane, a kid is crying, be sympathetic. It's not their fault. They got little itty bitty eustachian tubes. Inside the middle ear, we have three very small bones called ossicles. And I like to refer to them by their fun Latin terms, which have an acronym of MIS, Malleus, Incus, Stapes, or the hammer, anvil, and stirrup. And the function of our ossicles is to strengthen the wave function. Now, when the sound wave moves from our middle ear, which is air, to our inner ear, which is fluid, it's going to have a hard time. In the same way, if you were to jump into a lake or a pool or something, sounds kind of become muffled. It's hard to hear. That's because sound has a hard time traveling in fluid. And so to get our sound waves ready for the fluid of our inner ear, we got to strengthen them up. And that's what the ossicles are going to do. The sound wave is going to hit that tympanic membrane and send it vibrating. And the ossicles are going to move and push in the same way of that wave. And they're going to strengthen the wave. In the same way that you would push a kid on a swing and you would strengthen the wave function that they create with their legs when they're swinging back and forth, you're going to do the same thing with the sound wave. You're going to push it and strengthen it so it is stronger and ready to handle the fluid of our inner ear. So the vibrations from our tympanic membrane are going to move the malleus, incus, stapes, and they're going to hit in the oval window of our inner ear. Okay. So once we get to the inner ear, the bony labyrinth, this is where the magic happens. This is the structure for hearing and balance, and it's got a maze of bony chambers within our temporal bone full of perilymph, which is a fluid. We've got the cochlea, which is like a swirly shell shape, and this is where hearing lies. We've also got this vestibule, this like middle area. It's kind of unremarkable in terms of shape. And then our semicircular canals, which look like little horseshoe bones. The semicircular canals and the vestibule are for equilibrium, and the cochlea is for hearing. And all of them are little teeny bones bathed with fluid. So if we focus on that cochlea shape, we also call it the spiral organ of Corti. It is in the cochlear duct, and this is where the receptors are for hearing. And the receptors are hair cells, little teeny hairs, on a basilar membrane. And as the sound wave comes into our inner ear, it's going to move the fluid like a wave on an ocean. And that wave is going to move this tectoral membrane, which is going to pass over the hairs and bend them. Hair cells, once they're bent, that's what depolarizes the hair cell and starts an action potential that will move down our vestibular cochlear nerve and will eventually reach our auditory cortex in our temporal lobe right nearby. This is what that looks like. So this is our basilar membrane shown up. You can see those little teeny itty bitty hair cells here, and they're going to be moved by the tectoral membrane up top, which is moved by fluid. So fluid moves over it. Then it's going to move the tectoral membrane, which will bend the hairs. If the hair gets bent, it'll send an action potential down our vestibular cochlear nerve, which is going to end up in our temporal lobe, processed as sound. And the vibrations are going to move those hair cells. The information will first be passed to our brainstem, which will then eventually move to our auditory cortex. This is again what that looks like. Again, our basilar membrane with the hair cells. And what happens is the hair cells are next to each other and they're connected by a thin protein string, a thin fiber. And when the hair cells bend, that fiber that connects them are going to physically pull open a sodium channel. And sodium will rush in and depolarize those hair cells. We call this mechanogating. So these are mechanically gated channels that are responsible for our sound, for hearing. Okay, so I told you in the beginning that there were two parts of a sound wave that our ears need to process. We need to determine the frequency, whether something's high-pitched or low-pitched, and we also need to determine the volume. Frequency is encoded by which hair cells bend. So in your cochlea, you've got a wide array of sizes of hair cells, and they start really, really short, stiff hair cells, and they move to long, floppy hair cells. And high-pitched noises will only bend those short hair cells, and low-pitched noises will only bend the long cells. And so which hair cell gets bent will determine the frequency, will help your brain understand, oh, that's a high-pitched noise. Oh, that's a low-pitched noise. And this is what it looks like. So starting at our oval window, we've got short hairs for high-pitched noises, and it moves down as we move further into the cochlea. We've got long fibers for low-pitched noises. Frequency is encoded by which hair cell is bent, and volume is encoded by how long that hair cell stays bent. A really loud sound is going to keep the hair cell bent longer than really quiet sound, which will just bend it a little bit. So we encode volume based on the duration of the signal, and we encode frequency based on which hair cell moves. Okay, so that's going to be it for material for today. So we finished up vision, we reviewed how light travels through our eye, we talked about some common reasons why people need glasses, cool people need glasses. Then we switched gears, we started talking about our ears, we went through the external ear, the middle ear, the inner ear, and we talked about how our ears, our inner ear specifically, helps us hear frequency and volume sound. On Wednesday, we will pick up and we'll finish out the ears with a discussion of equilibrium. Again, not chemical equilibrium, the biology equilibrium. And then we'll switch gears and we'll talk about our last senses, smell and taste. Okay. I'm going to